Greetings. I will be presenting the paper entitled Effects of Ecologically Relevant Concentrations of Glyphosate-based Herbicides on Soil Nematode Community Structure. I'm Rowena C. Benavides from the College of Forestry and Environmental Studies of Mindanao State University, Maguindanao. With me are Dr. Reina Suzette Madamba, Dr. Nanette Sumaya, Dr. Ruben Amparado, and Dr. Genevieve Martinez from the Department of Biological Sciences of Mindanao State University Iligan Institute of Technology. So the soil is a habitat of millions of organisms. There is a very rich biodiversity inhabiting the soil. The productivity and success of our agricultural areas is anchored on a rich soil biodiversity. Soil also helps detoxify the pollutants and are essential for nutrient cycling. However, our soil, as our population increases, it leads to increase in the food demand, and this increases the intensification of agricultural activity that could lead to massive application of agrochemicals such as herbicides like Roundup. And these activities lead to changes in soil properties that could lead to a massive stress of our soil biodiversity. So in the graph, it shows that the use of herbicide has increased tremendously from 2011 to 2016. One of the heavily used herbicide is the glyphosate-based herbicide. However, our use of the herbicide increases, there is not a correlation between an increase in the amount of herbicide used and the increase in crop production. So the glyphosate-based herbicide, or the N-phosphonomethylglycine, in this paper, glyphosate refers to the commercially available Roundup. This is a broad-spectrum, non-selective, and post-emergence herbicide and one of the most prominent weed killer. This herbicide has gained rapid momentum because of the introduction of the glyphosate-tolerant crops. The glyphosate, or herbicide, is applied to fresh, actively growing vegetation. It is absorbed through the cuticle of leaves and stem of the plants and is transported by the phloem. This can block the activity of an enzyme that are vital for protein synthesis and plant growth. It has a process-specific effect on plants, thus it is generally assumed to be safe in humans. It does not persist since they are believed to be degraded easily by microbes. This is a photo showing the effect of a herbicide on the weeds. So on the you can see the weed applied with herbicides and without herbicides, that the destruction on the life of the weeds is very evident. So using glyphosate by itself repeatedly over time is the most important factor underlying the evolution of the glyphosate resistance. It becomes less effective at controlling some weeds, and the occurrence of glyphosate-resistant weeds affect crop production. Economically, it will impact the farmer's income, and they will need more amount of glyphosate with the declining effectiveness on controlling weeds, and increased use will also impact the environment and the human health. So the effects of glyphosate, once the glyphosate is sprayed on the leaf of the weeds, the chemical drain to the soil, causing changes in the microbial population, increase of fungal counts, and community structure of the nematodes. Records also show that it is harmful to mammals, earthworms, birds, tadpoles, and many other life forms, and freshwater carp showed changes in the liver cells and mitochondria. It suppresses the activity of the enzyme in a brown mucils. It may be used as a source of energy and nutrients by some soil microorganisms, increasing their numbers. It has also been associated with severe crop diseases. And considering the widespread use of glyphosate-based herbicides in the agroecosystem, their impacts on the composer communities and processes in agricultural soils, and its potential to be transported by wind and water erosion can contribute to the exposure of the herbicides to non-target organisms. So the objective of the study is to examine the effects of the ecologically relevant glyphosate concentrations on soil nematodes using a microcosm experiment, specifically to determine the responses of nematode community abundance and diversity to different ecologically relevant concentrations of glyphosate-based herbicides in a microcosm. So this is our microcosm. To assess the potential hazard of Roundup on the soil ecosystem, microcosm studies are suitable tool. The use of microcosm offer an excellent experimental system that offer the possibility to study the influence of specific aspects of contaminant behavior and ecosystem processes on communities under controlled conditions. It provides a balance between interpretability and practicability with ecological relevance in comparison. Field-based studies often are difficult to interpret because of the interplay of many ecological factors that may directly or indirectly influence the test organisms. So nematodes has been used as a bioindicator because, especially in a microcosm experiment, because they are found anywhere. They are taxonomically diverse. Some species are sensitive to environmental changes and some are tolerant to extreme disturbance. They play an important role in the decomposition and nutrient cycling, and they occupy several trophic levels. They also have short lifespan, making community responses to disturbances measurable temporally and spatially. So the methodology, soil collection for the experiment was taken from Maramag Bukidnon, Philippines, one of the food basket and thus a major contributor to the regional agricultural economy, from a field that is no history of glyphosate application or agricultural activity. Soils were then taken from the upper soil layer at a depth of 10 centimeter where nematodes are normally found in highest abundance. It was then immediately homogenized by gently mixing the soil and manually removing large debris. And then soil properties such as pH, organic matter, phosphorus, potassium, and nitrogen were being measured. The experimental design, 20 pots were arranged in a complete randomized design where 5 replicates per treatment was set up. These pots are filled with 5 kilograms of homogenized soils. So using the dilution of 3 liter per 300 liter of glyphosate and water per hectare, the treatments are as follows. This was extrapolated based on the above dilution. These were then applied once in the microcosm, and nematode collection and processing were done on the 15th and on the 45th day. To extract the nematodes, a tray method was used where soil was placed in a tray, and then after 78 hours, the nematodes were extracted. They were counted and then handpicked for identification. The results showed that there is no significant difference on the control and on the treatment per sampling periods, on the abundance and on the diversity of the nematodes. And so it follows that there is also no differences in the diversity indices and maturity index. The non-metric multidimensional scaling, you can see that the day 0 genus composition was very far from the genus composition of day 15 and similar with the day 0 and genus composition of day 45. You can see that day 15 genus composition, including the control, were overlapping each other, meaning that they do not vary in genus composition. So including the control and the with glyphosate treatments, the genus are similar. Hence, it is here the based on the similarity percentage, the average dissimilarity of day 0 and the day 45, including the control, was more or less 50%. And the dissimilarity will primarily caused by the decline or disappearance of the herbivorous Rotelenchus and Rotelenchulus and the predator Iotonchus. And also this dissimilarity was driven by the increase of the bacterivore Rhabditis. The nematode community structure for the sampling period, on day 0, it was dominated by herbivores, but on the 15th and 45th day, the dominance shifted from herbivores to the bacterivore-dominated nematode community. So in conclusion, the nematode abundance and diversity do not show differences with or without the application of ecologically relevant concentrations of glyphosate. So the used concentration is per is perceived to be safe on the nematode abundance. Thank you very much.